Most recently, scientists have managed to catalog all of the human brain cells and counted them up to be more than 3,000 different neurons or brain cells within the human brain. This initiative was one of the largest attempts within the scientific world where both biologists and neuroscientists and other scientists got together and put their own brains together to kind of map all these different neurons and brain cells that exist within the human brain. And this work is extremely important because it'll not only advance us in the neuroscience community, but also in the psychology and biology community. These scientists were able to go also very deep into very single cellular level and detect these molecular interactions among these single cells of neurons as well. It's also worth mentioning that this research was part of the NIH brain research, which is a huge institution. What these scientists did was to use tissue samples from donors who were deceased, of course. These scientists sequenced the RNA of 3 million individual cells gathered from 106 different locations within the human brain. And when the results came out and was published, even the scientists themselves were surprised by the number of different brain cells that they found to be existing in the human brain, many of which we were not aware of up until now. Another very interesting thing that they found was that the type of brain cells differed within the different regions of the brain. So for example, some neurons that were the same in the prefrontal lobe, they were not the same types of brain cells, aka neurons, in the back of the brain, for example. More importantly, they also investigated what type of neurons exist within the brainstem. Why this is important is that brainstem is one of the least researched area and region of the brain in the science. Scientists also looked at into the mechanism of gene regulations within the brain cells. And they found that the same gene within the same type of neuron could have different characteristics across the brain. For example, one gene could be turned on with one switch in front of the brain, but in the back of the brain, it could be turned on with another switch. In other words, and there were brain regional differences where switches could activate or block different gene expressions. What I just shared with you is obviously just the gist of the paper. I'll share the paper with you if you're interested in reading more about it. But definitely we're living in an exciting time because more and more advancement is coming in the science community and there's just still so much to learn.